lapel.
Learning her principles of biochemistry. Chapter 1: The Foundations of Biochemistry. Evolutionary Foundations, Part 1. Changes in the hereditary instructions allow evolution. Hey everyone, welcome back to our channel. Today, we'll be exploring how changes in our genetic instructions have paved the way for the incredible diversity of life on our planet. Let's start with the basics. Our genetic information is encoded in the DNA, the famous double helix structure you might have seen before. This DNA contains the instructions for building and maintaining an organism. Now, every once in a while, a change occurs in this genetic code. We call this a mutation. Most mutations are neutral or harmful, but some can actually give an organism an advantage in its environment. This process is what Charles Darwin famously referred to as natural selection, the idea that organisms best suited to their environment are more likely to survive and pass on their advantageous traits to the next generation. Imagine a gene being accidentally copied twice during DNA replication. This extra gene becomes a means for the cell to evolve, potentially leading to new functions and capabilities. But how did it all start? One hypothesis suggests that RNA, or a similar molecule, may have been the first gene and catalyst. This self-replicating RNA could have led to the emergence of the first cells. Think about it, billions of years ago, the Earth was a very different place. The atmosphere was devoid of oxygen, and simple organic compounds were relatively stable. In fact, a classic experiment in 1953 by Stanley Miller and Harold Urey demonstrated that organic compounds, including some amino acids, could be produced under conditions resembling the early Earth. As time went on, cells became more complex. The development of intracellular membranes and the acquisition of the capacity for photosynthesis and aerobic metabolism were key milestones in the evolution of eukaryotic cells. The linear sequence of nucleotides and DNA codes for the production of proteins, which then fold into specific three-dimensional shapes. This process is crucial to an organism's function and survival. Biomolecules first arose by chemical evolution. Ever wondered how life on Earth began? Well, it all started with biomolecules. In this video, we're going to explore the fascinating world of biomolecules and how they first arose through chemical evolution. Picture this: billions of years ago, Earth was a hotbed of chemical activity. Lightning, volcanic eruptions, and superheated thermal vents were the norm. These extreme conditions played a crucial role in the abiotic production of organic compounds, including amino acids and other simple molecules. In 1953, biochemist Stanley Miller conducted a groundbreaking experiment using a spark discharge apparatus to simulate the conditions of early Earth. The result? Formation of organic compounds, including amino acids, under primitive atmospheric conditions. But that's not all. Hydrothermal vents in the seafloor emitted superheated water-rich and dissolved minerals. These vents may have been the sites of early biogenesis, contributing to the formation of essential biomolecules. Now, let's talk about RNA. RNA, or ribonucleic acid, probably played a crucial role in prebiotic evolution. It acted as a template for the first genetic material, paving the way for the emergence of the first cells or protocells. The linear sequence of deoxyribonucleotides in DNA encodes the instructions for forming all other cellular components. This remarkable molecule carries the genetic information that specifies the structure of enzymes and other biomolecules. The sequence of nucleotides in DNA also codes for the production of proteins, which are essential for the structure and function of living organisms. These proteins fold into unique three-dimensional shapes, crucial to their biological function. RNA or related precursors may have been the first genes and catalysts. Have you ever wondered how life on Earth began? Scientists have been piecing together the puzzle of life's origins, and one of the key players in this story is RNA, or ribonucleic acid. RNA is a molecule that plays a crucial role in the transfer of genetic information and the synthesis of proteins in living cells. But what's even more fascinating is that RNA or related precursors may have been the first genes and catalysts in the evolution of life. RNA is made up of a chain of nucleotides, which are the building blocks of the molecule. These nucleotides contain a sugar, a phosphate group, and a nitrogenous base. The sequence of these bases in the RNA molecule encodes the genetic information, similar to DNA. But here's where it gets really interesting. RNA also has the ability to catalyze chemical reactions, acting as a biological catalyst. This catalytic activity suggests that RNA or similar molecules may have been the first genes and catalysts in the early stages of biological evolution. The idea is that in the primordial soup of early Earth, RNA molecules or their precursors could have formed spontaneously, perhaps near hydrothermal vents or through the action of lightning and high temperatures on simple atmospheric molecules. These RNA molecules could have acted as both carriers of genetic information and catalysts for chemical reactions, driving the early processes that eventually led to the development of more complex life forms. Today, our understanding of RNA and its role in the early stages of life's evolution is providing valuable insights into the fundamental principles of biology and the origins of genetic information. Biological evolution began more than 3.5 billion years ago. To understand the origins of life, we need to go way back in time. Earth was formed about 4.6 billion years ago, and the first evidence of life dates back to more than 3.5 billion years ago. Scientists have uncovered chemical evidence of life from as far back as 3.85 billion years ago, revealing the presence of distinct biological origins. Now, picture this: the terrestrial atmosphere at the dawn of life was nearly devoid of oxygen, and microorganisms were scarce. This allowed for the stability and eons of time needed for the improbable to become inevitable. Lipid vesicles containing organic compounds and self-replicating RNA gave rise to the first cells, or protocells, marking the beginning of biological evolution. These early cells arose in a reducing atmosphere and obtained energy from inorganic fuels, such as ferrous sulfide and ferrous carbonate. Over time, they gradually acquired the ability to derive energy from compounds in their environment and use it to synthesize more of their own precursor molecules. One significant evolutionary event was the development of pigments capable of capturing the energy of light from the sun, which could be used to reduce carbon dioxide and form more complex organic compounds. This process eventually led to the rise of photosynthetic oxygen-producing bacteria, known as cyanobacteria. As these oxygen-producing bacteria thrived, the atmosphere became progressively richer in oxygen, leading to a significant shift in the evolutionary landscape. Some lineages of microorganisms gave rise to aerobes that obtained energy by passing electrons from fuel molecules to oxygen, taking advantage of the energetic benefits of aerobic metabolism. Today, modern bacteria and archaea inhabit almost every ecological niche in the biosphere, showcasing their remarkable adaptability and diversity. Through the process of biological evolution, organisms have acquired the ability to use virtually every type of organic compound as a source of carbon and energy. Fast forward to about 1.5 billion years ago, and the fossil record begins to show evidence of larger and more complex organisms, probably the earliest eukaryotic cells. These cells underwent several stages of evolution, acquiring the capacity for photosynthesis and oxidative phosphorylation from endosymbiotic bacteria. The genetic foundations of biological evolution are deeply rooted in the structure and function of DNA. The linear sequence of nucleotides in DNA encodes the instructions for the formation of all cellular components and provides a template for the production of identical DNA molecules to be distributed to progeny. The structure of DNA allows for its replication and repair with near-perfect fidelity. Before a cell divides, the two DNA strands separate locally, and each serves as a template for the synthesis of a new, complementary strand, ensuring the continuity of genetic information. The study of biological evolution has led to the classification of organisms into three distinct domains of life: bacteria, archaea, and eukarya. Each domain represents a unique branch in the evolutionary tree of life, reflecting the remarkable diversity and unity of living organisms. So, there you have it. The story of biological evolution is a journey that began more than 3.5 billion years ago and continues to unfold to this day. The intricate processes and mechanisms of evolution have shaped the world we live in and continue to inspire awe and wonder. Thanks for watching. Be sure to watch part 2, like, and subscribe for more videos on the world of biochemistry and the origins of life. See you in the next video.
Pharma Pal.